Hundreds of macroscopic effects that we encounter in everyday life find their origin in the behavior of organic molecules. Linked transformations of organic metabolites diminish and amplify one another within the human metabolome, the ensemble of chemical reactions that take place within the human body. Organic molecules that emit light in response to an electrical stimulus form the basis of organic LED technology. Organic polymers, consisting of long chains of repeating units, intermesh to produce emergent material properties that we rely on. In all of these applications, a molecular level effect, one that depends only on the behavior of a small number of imperceptibly tiny molecules, produces a macroscopic response we can observe. The goal of the Molmodac project is to describe molecular level mechanisms like these using extant chemical literature and the language and tools of Chemistry 332. To begin the process of topic selection, I want you to close your eyes. Close your eyes and imagine a macroscopic effect that you observe daily. Anything from brushing your teeth to driving a car. Pause the video and take a few seconds if you need it. Before moving on, have a macroscopic effect in hand. Don't worry about whether or not the effect's origin is even chemical yet. Do you have an effect in mind? As an example, I'm interested in how neurons in the skin transduce the pressure of touch into electrical signals. With this effect in mind, I next ask myself, what is known about how this effect works at a microscopic level, and how is it known? To learn this, I need to turn to experts on the subject and the neurological literature. Experts on the internet may inform you how a mechanism works, but they'll rarely describe how that knowledge came to be known. Thus, an important part of Molmodac is interfacing with the primary chemical literature to discover the experiments that generated knowledge of a mechanism. A quick Google Scholar search reveals that specialized neurons called sensory neurons are responsible for the transduction of physical stimuli into electrical impulses. In fact, the first hit on Google Scholar is a paper that describes how sensory neurons encode pressure into electrical impulses. As you browse the literature, look for keywords that suggest a mechanism operating at the level of a relatively small number of atoms. Small enough that you could envision all the important pieces in a single JMOL or Marvin sketch window. Try a quick Google Scholar search of the effect that you thought of earlier. Pause the video and resume when you have one or two references in hand. Remember to keep an open mind as you search. You may need to try several different phrasings of the same idea. Now let's dig into this first reference and look for evidence that a molecular level mechanism has been studied. At this stage, you'll need to look for keywords that suggest the importance of molecular structures. In this particular paper, the importance of specific cRNA and mRNA, as well as a protein receptor, P2Y1, is described. As you browse the vast sea of literature for information about your topic, look for key proteins or reactions in which the compound is involved. For the sense of touch, the P2Y receptors appear to be critical. I've tracked down a paper describing in detail the steric and electronic properties of the ATP binding site in this protein. Notice that at this point, I've zoomed in from the macroscopic effect of the sense of touch to a microscopic mechanism, the binding of ATP to P2Y receptors. Try doing the same for the macroscopic effect that you've chosen. Don't worry if it's organic or even if it's chemical in nature. Just look for evidence that researchers studying the effect have zoomed in and looked for deeper causes for the large-scale phenomenon. The idea of zooming in is a good analogy for the Molmodac topic selection process. Keep in mind that because this is an organic chemistry course, the microscopic mechanism you focus on should fall within the domain of organic chemistry. But aside from this requirement, the choice of topic is up to you. In the next webcast, I'll talk a little about the primary literature collection and how it can help you jumpstart the topic selection process.